Hello, everybody. I am Aleksei. I am working at the National Institute for Medical Research, but now I will talk about my previous research, which I was doing during my PhD studies at the EMBL in Heidelberg. I'm a bioinformatician and do only computation stuff. I don't do experiment, but yeah. So in nature, most bacteria actually exist in communities, and it's rarely you can find a pure culture. And microbes are very important for our ecology. They can be found in all the niches of our planet, from volcanoes to the deep oceans and the cold polar ice caps. They, for example, could produce food for us. They digest food. They clean waste and pollution. And for example, in human, there are 10 times more bacterial cells than as its own cells. And what's also, if you take the dry weight of the human gut microbiota, it will be around, it's more than the liver weight. So it's like a separate organ, which is important in digestion. So in this simple example, this bug thermite, it has inside cellulose degrading community, which can basically take the lignocellulosic material and degrade it to more similar compounds, which can be absorbed by the bug, but overall it doesn't do on its own this stuff. So we can nowadays the technology exists, which we can basically sample environment and extract the DNA from it, do sequencing, and then from those sequences we can understand what kind of microbe was there. And depending on the environment, there are very different. The compositions of environments are very different, and sometimes in the very extreme environments you can have a very small number of species, but in the environments like, for example, sediments or human gut, you can have up to 1,000 species. And ideas that we try to understand using computational approaches how basically their metabolism is functioning. And because metabolism there is a large belief that what the shape of this community is one of the driving forces for shaping the community structure is metabolism. So the way how, despite that there can be different bacteria found in soil, lake, and human gut in different parts of the world, irrespective of that, sometimes you can find groups of bacteria which are always together. They find statistically you can find them more often as a group of species, but not separately. And that can indicate that they might somehow functionally related, because irrespective, as I told, irrespective of where they come from, you have always this kind of subgroups of species which are found together. So basically the hypothesis is that strongly interacting species will significantly co-occur if they have a kind of a functional relationship. So if you take the samples from different Earth places, you can identify species from the DNA sequencing, and then you can basically quantify, do some statistics. Basically what you calculate is that how often they found together than alone, and you can find a group of 2, 3, 4 etc. members, and basically it will build some kind of sub-communities in the sample. So they are always present in many samples, and they always present together but not individually. So, and then you can model those, like model metabolic properties in those communities using some flux balance models. I will explain how they work a little bit. And then contrast them against random assemblies, which you would not, basically against your expectation. What if you would be, if you just randomly choose them from the other population of bacteria. So important that to study that important to expand it from 2 to 3 to 4 bacteria, because sometimes if you do just statistics on 2 bacteria, you cannot infer the higher interactions, and moreover, it can be some misleading because you don't know whether the AB was present because of A actually, but because maybe there was another bacteria which was making this interaction. So yeah, it's a bit confusing here. But if you look at this, so we identify from all these samples of DNA sequencing, we identified these groups of bacteria like of size 2, 3, and 4, and there are nearly 7,000. And if you look at the species composition inside these different groups of co-occurring communities, you can find that the overlaps are very small. Well, you cannot see here, but the overlaps overall small. That it means that there is so that you cannot everything infer only from pairs, but you need to look at the broader picture. And despite that you have only three dominant phyla, actually you can see here they are. So it's like a plot diagram, like a circle diagram, where you can see that each ribbon represents that if species from this phyla of bacterium are more often found in this one phylum, and why it looks so somehow strange. Anyway, so what you would expect that they are more that the interphyla interactions are co-occurring more often between the phylum than just inside the phylum, and that's actually from evolution it's quite obvious, but yeah. So now we want to model these communities. So basically, for each of the species, you can reconstruct metabolic model. So what is metabolic model is just a set of whole possible reactions in that species, and you can curate them for some transport reactions and so on. But per species, you can have around 1,500 reactions, and then you can put them together to communities and model the properties how they interact. So I don't know why it's a bit yeah, wrongly displayed, but somehow it should be better quality. I don't know why. So anyway, so you can represent compactly the metabolic model. You can present as a basically a set of equations, linear equations, and solve this so basically you have per metabolite you have a mass balance equation where you use it for all the reactions where it was synthesized and degraded, and you assume that there is a constant growth steady state condition, no metabolite change over the time, and it boils down to solving this system of equation. Usually you have more metabolites or more reactions than metabolites, and then you do some optimization to find the flux distribution, which feasible flux distribution, which is can be under those conditions in the cell. And this approach calls flux balance analysis. So basically you optimize for growth, and growth is just a reaction where you put the all possible compounds like needed for protein synthesis, RNA synthesis, lipid synthesis, and so on. So what you can get out of it is a flux distribution under some given condition, intracellular flux distribution. So what it is useful for example, you have experimental measurements. You measure your glucose uptake or some secretion rate, and what you can do, you can predict quite well growth or ethanol secretion rate, at least for model organisms like Saccharomyces cerevisiae or E. coli. This is very quite has good accuracy. And in this case, for this study, I was using it for example, you can estimate the minimal growth requirements for a given bacteria. Basically, what kind of compound should be present in the minimal in the medium in order to support growth of the species. And there is a famous natural basically Darwin's naturalization hypothesis, which implies that so that co-occurring species must be likely metabolically dissimilar due to their kind of a risk of competitive exclusion. So basically, you cannot, if there is something in the habitat, you can't reduce similar species because it will be outcompeted by the same guys there. So, and this basically we try to answer this question by using the metabolic modeling. And the first thing was to check basically what you do for each of for all these identified communities of 2 pairs, 3 species and 4 species, which I was showing. What you can calculate phylogenetic distance of that community. And then I don't understand why this should be, well, you can see this line, right? So basically, what you can quantify the phylogenetic distance of the species between the species inside that community, and then contrast against the random control. The random means that those were just randomly picked in a sample community, and you quantify the same metric, like phylogenetic distance in this case. And the red line is only for co-occurring community. You can see that there is some significant differences between distributions, but overall it's a very weak signal, and you cannot really say that okay, phylogeny completely describes their co-occurrence. So then was the idea, okay, can we measure the growth requirements of this? Can we not measure, but estimate the growth requirements based on their models? So basically, and compare them across in these co-occurring communities. So basically, we can estimate from modeling how similar are their metabolic requirements. And in this case, if that one needs these 2 molecules and the second species needs other 2 molecules, and that the common of them is 33%. So basically, we can quantify from the stoichiometric modeling, you can quantify this metabolic resource overlap, and this is a kind of a measure of competition, how similar they would, what kind of resources, the similarity of their minimal needs. So, and you would expect that in closely related species that they are evolutionary very well similar. You would expect that this overlap would be big, and for the very distant species, the overlap would be small. And just as a validation, you can see that each dot in this is a community, and you just compare this with the phylogenetic distance between those species in that community and this metabolic resource. You can see that the models actually can capture evolutionary signal, so that for example, the closely related species have complete overlap of resources, and then the idea was, okay, can this metabolic resource overlap explain their co-occurrence? And you can see that the same thing for each density plot demonstrates basically the communities of 2, 3, 4 species, and this is the red is a signal only for the co-occurring communities, and the gray is the random control. And again, it cannot really very well describe their co-occurrence. And then the idea was, okay, what if we take, for example, not those co-occurring communities, just take the whole sample, all the species there, and they calculate the same property inside those samples. And then this is a different samples with different amount of species, and you could see that overall that there is a kind of a presence of competition indicated by the higher metabolic resource overlap in those communities. So basically, they likely to the samples in the sample species are likely to have the same use the same resources, which is kind of a description of the habitat, which is more or less the same that they compete, which is logical. However, so imagine that sometimes the environment changing constantly, and and it would be beneficial for species, for example, if when they cooperate, they could survive potentially on a smaller amount of nutrients. In this case, you see the two communities, one is non-interacting, another one is interacting. They more or less have similar, well, different a little bit requirements, but what it says basically, that if community would not interact, it needs 4 metabolites in order to all of these species would grow there. It needs 4 external metabolites. In other case, this would need only 1 metabolite. So potentially they could exchange these 3 metabolites just by if they could complement each other. And if you take the all species from available sequence species, you construct metabolic models for all of them. And then what you do, you randomly sample them, assemble, and calculate minimal growth requirements. Well, if they were interacting and then they were not interacting, and you can see that there is a high potential for interactions. So basically, we try to come up with a metric to measure the potential of metabolic interaction inside those communities which were identified from this data. And you can see that for the sample, if you again calculate this metabolic interaction potential for the samples, you can see that they don't really like to kind of, at least this is significantly lower in many cases, irrespective of the size of community, that they less likely to metabolically interact inside this habitat. So kind of they even might be, I don't know, independent in terms of, yeah, so they again compete. So, but however, if you calculate the same property, but for those which were co-occurring, again, something, so this red line is a co-occurring communities. For, again, you have pairs, triplets and quadruplets, and the gray line is a random background. So you just not those co-occurring, but you just pick up from the same sampling space, just random assemblies, and you calculate the same property. You can see that this is much stronger, which were compared to the, for example, phylogenetic distance or their similarity in their requirements. So there is kind of a cooperation potential which makes them keep together, perhaps, I don't know. So then the next question was to answer, are those interactions inside those communities, are they of mutualistic, for example, nature, or commensal, and from which metabolites those interactions are happening? And for that, we developed another framework called metabolic species metabolic coupling analysis. Basically, what it can say, it can say the importance of exchange in the community between species for a given metabolite in order one of them would be growing. So, and then you can score each metabolite and each species, how they important are in the whole network of communities. And then just it's now explained, we came up with a simple scoring system, which is uptake score and species coupling score. So uptake score usually is needed because sometimes there are many alternative pathways. So basically, for example, organisms can utilize different carbon sources, and you need account for that. So basically, this framework allows to enumerate all possible metabolic interactions through the whole like complete genome scale models, and in the community scale, it can enumerate all possible metabolic interactions, and it can say, for example, from all the solutions, how much, for example, metabolite M1 was needed to grow for species S3 in this case. And in this case, is 0.5% of time because there is alternative in this case, M2. And then what typically in the community of, in this case, is a kefir drink community, it's mostly made of different lactobacteria and yeast. And you could see that it would require all these compounds, one of these ones, and one of these ones. So this is just like practically how it looks, the solutions. And yeah, these compounds are needed for this to grow. And And then the species coupling score, because also the compounds can be produced from different species. You can need to evaluate which species were giving, or who was the donor in the community, and if it is a crucial member of community, you can, for example, expand it and use for if you want to do some kind of targeted approaches where you want to remove or add the most crucial member to the community, so that to keep them together. And in this case, and the method was, there is no much experimental data measured on the metabolic exchanges, but those which are available for microbes, like where they can measure the transfer of metabolites between species, it captured all these exchanges. And so basically, the higher the score, the more important metabolite is. And we calculated that, again, the same for the co-occurring communities and for random control. You can see that this species metabolic coupling score is much stronger in these co-occurring groups of species, and that means that their metabolic interactions are more important there. And if you look at the metabolites, mostly, well, it's just if you just sum up all the amino acids and all the carbohydrates, this is in the log scale. So there were, for example, 4 times more amino acid exchanges absorbed than in random controls in those co-occurring species. And if you look how the exchanges were looking like in this graph of species, they were enriched in mutualistic interactions. So then we came up, we wanted to see whether this, in principle, this cooperation of exchanges of metabolites can exist in some cultures. And we took the 4 bacteria and 1 yeast, and then propagate for 10 days and measure the measure the basically who was present in the final culture, and under 2 conditions. One was the rich medium, where many compounds were present, and another one where just almost randomly removed 13 amino acids. It was just a group of amino acids which was dissolved in the medium during the preparation of species. So we were not like selected somehow, just any metabolite amino acid could be. You could see that in the monoculture, all of them growing in the rich medium, but in the poor medium you cannot detect the 2 species of lactose. And this is a reproducible experiment, repeated many times. So basically they don't grow. However, in co-cultures you can see in rich medium they grow in all, but in the poor medium they grow on. So there is a potential metabolite exchange between those species. And then so in nature it also in principle can happen. And then in the same, if you apply this modeling framework, you can see that what are the metabolites, for example, and currently we're working on the demonstrating those actually in this community to detect them, whether these are metabolites, what we predict, and what we can absorb. So overall, this study was more about like basic how to understand the role of metabolic interactions for species co-occurrence, and we could see that competition actually is dominant at the sample level overall in the habitat. However, for cooperation, there is this metabolic cooperation in the co-occurrence. Somehow it is important, and if you quantify the effects of competition again and cooperation is much higher in those co-occurring subcommunities. And basically, so the idea is that the cooperation kind of promotes the coexistence among these habitats. Yeah, I'd like to say thank for the previous group I was working with at the EMBL, and this is my current group I'm working. And you for attention. Yes.
you may have explained this, but I didn't understand. But so I'm not quite sure exactly what you mean by cooperation here, because it seems that you're defining cooperation in terms of co-occurrence and similarities. But it seems to me that it could be the same thing as parasitism. So it might be that the one species benefits from the presence of another species because of the metabolites created by one species.
Yes. So I defined co-occurrence. I defined as the species which co-occur among them. Well, from statistics of basically if you can observe the species if they are co-occurring in groups more often than on alone, and by cooperation we try to find the metrics for the cooperation, and one of them was this metabolic interaction potential, and which was this basically. Basically, it's a difference between the requirements if they were interacting and if they were not. So and however, and about and this metric, for example, in those co-occurring communities, this was much stronger than in the random assemblies. But and this is a cooperation measure, because you meant about the competition, right?
Well, I mean that cooperation is mainly what I'm interested in. So could you explain it in more detail?
Yeah. So idea is that imagine if you have two species, right, and one can have something can synthesize that another one can take up, and vice versa. And if they have a third, for example, metabolite which were needed for both of them, if they were not cooperating, you would need two metabolites in order each of them to grow. For example, in this case, this community takes this one, and then that one takes the yellow one, and this one. So basically, they are not interacting, but from environments they need at least 4 different compounds in order all of them to grow, right? And if but on the other hand, this community which is cooperating, it can also give the metabolite to that one, so that it doesn't need it to take from an environment. And in total, for all of them as well the same. So basically, externally it only need to take only one metabolite. So the difference in those would be 3 metabolites, and this is their kind of metabolic cooperation potential. So how much this community could gain by cooperating.
Yeah, what's not clear to me is how this distinguishes from parasitism, in the sense that you also have the case that if you have a parasite in the host, the parasite needs something which the host provides.
Yep. So for that reason, you calculate their overlap of basically needs. So, and this is what the overlap of the needs is. Basically, if you have, this is a bit different. You can quantify what are their similarities and their needs in this case. For example, in this case it would be X, and the 33% of their needs.
Right. If you go to your last slide, it seemed that the flow of nutrients seemed to be in one direction. There was no kind of there was one species which provided nutrients but didn't receive any.
It has of course the environment. I didn't display the environment. There is always something goes in the environment to each of them, and this is just that environment lacks something that this one can provide them those.
Yeah, but it seems to me that the one at the top right isn't cooperating with the others. The others are parasites on this Cerevisia.
Well, not necessarily parasite. The thing is that then Cerevisia grows. It knows that it leaks out metabolites. It's basically all that. It's like they just by diffusion or by something else.
Yeah, sure. Okay, but I mean the point is that it's not benefiting from the presence of the other species.
It may benefit if the compounds are inhibitory. Product inhibition, like ethanol or so. Okay. If another bacterium eats the ethanol that Saccharomyces excretes, then it could benefit.
Okay. So does this analysis take account of those kinds of interactions?
Well,
it could. Or I'm not sure if it
yes,
takes here, but it could.
Yes, yes. I mean, so in this case, those are not needed for that under these conditions for the like sufficient growth. This is what it means. So it doesn't harm for it to if you kind of because it is difficult to quantify it, but of course overall maybe there is some sort of in terms of I don't know parasitism, but I don't know how it's also can be called commensals, right, where they don't really harm.
Yeah, sure. But I think that being commensal is very different from cooperation, and the dynamics of cooperation is very different from the dynamics of being commensal.
You mean that there is no like you mean that there is no reciprocal mutualistic interaction?
So mutual interactions, cooperation is something which can stabilize coexistence, whereas these kind of interactions, at least if those arrows correspond to the what affects what, then there's actually none of those dynamics actually stabilize the
Yeah, I understand what you mean, but this is first of all it's experiment and this was more to show that there can be potentially that you can as a group can benefit from the from the metabolic interactions, but as a group it benefits because you can see that they cannot survive on alone. So only when they all together. But in terms of cooperation, what I meant, for example, in those co-occurring communities, the graphs were, for example, they were enriched in the mutual interaction. So they were species which were exchanging the both ways. But I know for dynamical systems, you need something to at least two connections to make it stable.
Much of the scores relied on the number of metabolites. But I can imagine, for instance, that species exchange or cooperate on the basis of 10 different carbon sources, but the exchange is very weak. Perhaps they compete for only one substance, but perhaps that competition for that one substance is very fierce.
Mm-hmm.
So how can you translate this into your analysis?
So yes, so in all those the data which you can take in from environment usually is a snapshot of certain point in time, somewhere in autumn or somewhere in spring, from some lake or something. So there are many data points. So overall, of course, there is always competition at the specific sampling site for all the nutrients. But overall, if you can see somehow they exist, these species present in all those environments, and maybe at one time point they compete for it, and then in another time point someone just can just make them grow as well, somehow provide them benefit, and overall in time maybe this is how evolution somehow evolved them all together to live in such a way.
Yeah, that's all very clear, but I think in addition to the number of metabolites in which they overlap, you should also have some information on strength.
Yeah, of course.
Right?
Of course, I can quantify all of this, but you need to know if you don't have really a growth rates, because I don't know at what extent they are growing. I know that what is the effect of, for example, glucose on one species and glucose on the other species. However, in reality, I don't know how much they grow. So the importance I know of all of these metabolites, if I would assume that they're equally well grown, this I can of course quantify, but I cannot translate it for example, if I have only sequence data, I cannot get this type of information. But yeah, of course it has to be accounted. But if you know the conditions, for example, in this case, if you know the conditions, you can pretty well simulate the interactions, and yeah, it depends, and even you can expand it to dynamics. So basically, you would do it per time point and could see what is happening. Finding all possible solutions at a certain time point, and
Now
then feeding
you
it.
speak about time points, what about space? So this analysis is probably with some homogeneous.
Yeah, well, it is assumed heterogeneity, homogeneity.
So
Sorry.
I would like to see it in a gradient system,
Wow.
dimensional, or bacteria interacting with each other in space, exchanging metabolites, like in biofuels, of course. Maybe Rosalind is doing some of these or others.
Well, theoretically, you could make a space structures and define physical property for each
So
structure.
if you want to solve this system of non-linear or linear equations, algebraic.
It's a linear equation.
Linear, okay, so it's in one point, so you can make it in different points then, but you have to include diffusion.
Well, if you can from input to the system can be your dynamic model, like something is changing, and then I recalculate everything according to the input system, and then
I think things would change if you put space in.
yeah, of course, it's just they would definitely, but yeah, it's very simple, but
just to follow up on that question, actually, the paper you cite by Holman Murray in Science,
yes,
they show that basically if you agitate the system, it's an algae yeast symbiosis mutualism. If you agitate it, you break down the nutrient exchange. So they show the importance of things like physical associations and just
so they also, in that case they demonstrated that there is exchange of nitrogen. I don't remember from what, and in that case it's just this system was, and the condition in which they were growing, it could capture that nitrogen. This is and that specifically I don't remember algae, no.
Basically,
yeast to algae,
algae could convert nitrite to nitrate, which the yeast can use, and the yeast were converting glucose to CO2, which the algae could use. They show that it breaks down when you agitate the system, and the point was that physical associations were important between the algae and yeast.
Yes, but they had just measurements. That was the point. My point that it was not about particular community, but just like you could capture the metabolic exchange. For me, it was the point.
Okay, I see.
From these metabolic models under those conditions. Yes.
Hi.
Hello.
I have a different question. So think about these co-occurrences. Why do they occur? Then, theoretically, I would say if I was a bacteria, I would prefer to be able to do everything by myself, so I don't depend on anybody else. So, but in nature, it does occur. So, in your point of view, why?
Well, thing is that it's not like that, unfortunately. And you have we don't do everything. We do it like for example, in that environment. The bacteria usually tend also evolutionarily to lose the genes when they go from one environment to another one, simply because it doesn't need. So it doesn't want to keep up everything. So, and sometimes they're mixing and they can have a mutual benefit. So in evolution, you don't have everything. We cannot do everything.
It's a matter of efficiency.
Yeah. So
in the end, how do you allocate your resources? If you have this substrate, you must be able to eat that, and then you develop an organism which can eat that substrate.
yeah, and then if you just don't change the substrate at some certain time in time, you will lose those genes because there is no evolutionary pressure on eating that. So you don't do everything on your own.
So, if I understand correctly, so that's the result of a random mixing, some sort of results. You evolve independently and you lose some opportunities in the gene disappear, and then randomly they meet with each other and they found that by working together they can survive better.
Overall, yes. From the global picture, perhaps yes. So overall, there is always kind of competition in the same room, but maybe just what keeps us together is the friendship, right? And again compete in another room. But
Have you seen about by working together, they can actually use the resource better? More efficient?
they not more efficient, but sometimes I can give you something, you can give me something back, but then the time comes that I cannot use what you have, but you can produce something, and I will use it. So that's the kind of they complement each other by biosynthetic differences. But yeah, I don't know. It's just what we observe. So
I think there might be a potential of share. Coming from sort of a chemistry background, I'm thinking that by sharing the burden of, say, the biosynthesis, if they don't do it, if individuals don't do everything by themselves, they might save the driving force, they don't lose the energy during biosynthesis. But let me think it clearly. The idea just popped out when I'm hearing it, so we can talk later.
yeah, but yeah, I don't know the. Yeah. Any other questions? Thank you very much.